Okay, so we're about to dive into the world of a true master manipulator. A tiny, single-celled organism that is so incredibly cunning, it manages to turn our own body's elite defenders, the very cells designed to hunt and kill invaders, into its own personal hideouts. This is the story of Leishmania, a parasite that's responsible for a truly devastating range of diseases all across the globe.
So here's our roadmap. First, we'll look at the real human cost of this parasite. Then we'll get into its absolutely fascinating double life. We're going to unpack the genius strategies it uses to outsmart our immune system, get a handle on the massive global scale of the problem, and then finally explore the ongoing battle to fight back against this really formidable foe.
Right. Let's start with the impact. Because before we get into all the cool, intricate molecular biology, it's so important to grasp the very real and often tragic human cost of leishmaniasis.
You see, the disease shows up in several pretty devastating forms. There's the cutaneous form, which causes these disfiguring skin ulcers that can leave permanent scars and create a huge amount of social stigma. Then you've got the mucocutaneous form, which can horrifically destroy the soft tissues of the nose and mouth. And then there's the visceral form, often known as kala-azar, which is a systemic and frequently fatal infection that attacks internal organs like your spleen and your liver.
And what causes all of this devastation? A single-celled protozoan, completely invisible to the naked eye. It's just a wild reminder of how a seemingly simple organism can unleash such profound and complex damage on its human host.
So let's lock in the official definition here. Leishmaniasis is a vector-borne disease. The two key terms you really need to remember are vector, in this case that's the sandfly that carries it, and obligate intracellular, which is absolutely crucial. It means this parasite has to live inside one of our cells to survive and multiply. It's completely dependent on hijacking our own cellular machinery.
Okay, now let's get into the parasite's remarkable biology. Leishmania is basically a shapeshifter. It leads this kind of double life, taking on two totally distinct forms so it can thrive in two completely different environments, the gut of an insect and the cells of a mammal.
And just look at the contrast here. On the left, that's the promastigote form. It's long and thin, with that whip-like tail, the flagellum, that lets it move around, perfectly adapted for life inside the sandfly. Then on the right, you've got the amastigote, small, round, with no external flagellum. This is its stealth mode, the form that replicates inside our cells.
So think of it this way. The promastigote is the infective stage, the one that gets injected into us by the sandfly. Once it's inside our body, it cleverly triggers its own uptake by our immune cells, and then it transforms into the amastigote, which is the replicative stage. These two forms have fundamentally different structures and biochemistries, each one perfectly tuned for survival in its own specific niche.
Let's just walk through this two-host or digenetic life cycle. It all starts when a female sandfly bites an infected mammal and sucks up macrophages that are just packed with amastigotes. Inside the fly's gut, these transform into the motile promastigotes, they multiply, and then they travel up to the fly's mouthparts. When that fly takes its next blood meal, it injects these promastigotes into a new host. They're quickly swallowed up by our macrophages, where they switch back into amastigotes, multiply like crazy until the cell bursts, and then go on to infect even more cells, keeping the cycle going.
This diagram really shows you just how complex these single-celled organisms are. I mean, they're far from simple blobs. Take a look at the single, large mitochondrion, and inside it there's this unique structure called the kinetoplast. It's this dense network of mitochondrial DNA, a real hallmark of this particular order of protozoa. All of this intricate cellular machinery is just fine-tuned for one purpose: surviving inside a hostile host cell.
So this brings us to what is, for me, the most fascinating question in all of Leishmania biology. How on earth does it survive, and not just survive, but actually thrive inside a macrophage? A professional killer cell of our immune system whose entire job is to eat and destroy invaders like this.
Well, one of its most brilliant tactics is the Trojan horse strategy. Instead of going head-to-head with a macrophage, the promastigotes that get injected first infect neutrophils, the immune system's first responders. Inside this temporary shelter, the parasite starts to transform. Then, the infected neutrophil undergoes programmed cell death, or apoptosis, and gets quietly cleared away by a macrophage. This is how the parasite gets delivered directly into its target cell without setting off any alarms. It is a truly silent and stealthy invasion.
And once it's inside the macrophage, Leishmania unleashes this sophisticated molecular toolkit to take control. A key weapon is a surface protein called GP63. It acts like a pair of molecular scissors, chopping up host proteins like mTOR to dismantle the cell's defensive pathways. It can also switch into a slow-growth, stringent response, basically going into low-power mode to avoid stressing out its host cell, which is perfect for setting up a chronic long-term infection. And if that wasn't enough, it has a remarkably unstable genome that lets it adapt quickly, and it has the ability to become completely dormant or quiescent, which makes it invisible to our immune system and resistant to drugs.
So, now that we've seen how this parasite operates at the cellular level, let's zoom out and really appreciate the immense global health challenge that it represents.
As you can see from this map, leishmaniasis isn't some niche tropical disease. It's endemic in about 90 countries, spanning Asia, Africa, the Americas, and even Southern Europe. You can see the pretty distinct geographical spread for both the visceral and the cutaneous forms of the disease.
And the number of people living in these areas is just staggering. An estimated 350 million people are at risk of contracting leishmaniasis. That number alone places it firmly among the world's most significant parasitic diseases.
Every year, there are up to 1 million new cases of the cutaneous form. And while that's incredibly debilitating, it's the visceral form that is truly terrifying. Even though it has fewer cases, around 90,000 a year, it carries a shocking mortality rate of up to 95% if it's left untreated.
And a major complication for any control effort is that leishmaniasis is a zoonosis. It circulates in animal populations. In many parts of the world, especially around the Mediterranean, dogs are the primary reservoir hosts. This makes trying to eradicate it incredibly complex, because you're not just fighting the disease in humans, but also in a huge and very mobile animal population.
So given how complex this parasite is and the sheer scale of the problem, what tools do we actually have to fight back?
Well, just diagnosing it presents a whole set of challenges. The gold standard is trying to grow the parasite from a biopsy, but that's slow and really difficult. Microscopy is faster, but you need a highly trained eye to spot the parasite. Serology, which looks for antibodies, can't always tell the difference between a current infection and one you had years ago. And while molecular methods like PCR are super sensitive, they require lab equipment that simply isn't available in many of the resource-poor areas where this disease is most common.
The treatment picture is just as challenging. The traditional antimony-based drugs are really toxic, and we're seeing widespread resistance. Newer drugs work, but their effectiveness varies a lot between different Leishmania species. And here's a fundamental problem. For a lot of these drugs, we still don't fully understand how they work. The Holy Grail, what everyone is searching for, is a safe, effective, short-course oral drug that works against all forms of the disease.
But there is a glimmer of hope here, and it actually lies in our own immune system. We know for a fact that people who recover from an infection usually develop a really robust, long-lasting immunity. The proof is in the historical practice of leishmanization, where people would actually deliberately inoculate themselves with gunk from a lesion on a hidden part of their body just to prevent getting disfiguring facial sores later on. It sounds grim, but it tells us, in principle, a vaccine is absolutely possible.
So, why don't we have one? Well, the path is anything but simple. Live attenuated vaccines, using a weakened version of the parasite, show the most promise in lab experiments, but there are major safety concerns. Given how plastic Leishmania's genome is, there's a real risk the weakened parasite could revert back to a dangerous disease-causing form. The simpler subunit vaccines, made of just bits of the parasite, have so far failed to provide enough protection. And on top of all that, there's a serious lack of investment from major pharmaceutical companies, because at the end of the day, leishmaniasis is overwhelmingly a disease of poverty.
And this leaves us with a final and pretty provocative thought that goes way beyond the lab. In many of these endemic areas, a huge portion of the population might be asymptomatically infected. They're carrying the parasite, but they don't have any symptoms, and that exposure likely gives them immunity. So here's the question. If we developed a perfect drug that could completely clear every single parasite from every person, would we risk making the entire community immunologically naive and vulnerable to even more severe outbreaks down the line? It's a really complex, thorny question that forces us to think about the delicate balance between the parasite, the host, and the entire community.